Hello students, welcome to EPG Pathshala. I am Dr. Renu Tyagi from Department of Anthropology, University of Delhi. Today we will be discussing the module Formation of the New Population and Species from the paper Human Origin and Evolution. Now let's see what are the objectives of this module. Number 1 is to understand the concept of the speciation. Number 2 is to study the isolating mechanism driving speciation and to gain the insight into the macro and microevolution. Let's understand the concept of population and species. The conceptual framework of the evolution is that it is central to the development of the life forms, including the mankind on earth. New life forms develop from the pre-existing ones by incorporating the modifications for variations, and this evolution is referred to as the descent with the modification. In order to understand the evolutionary changes which leads to formulation of the new life forms, the basic unit of evolution must be studied. A population constitutes the community of the potentially interbreeding individual in a specific area at a certain time. Dobzhansky in 1955 emphasized that Mendelian population relates to a reproductive community of sexual and cross-fertilizing individuals sharing the common gene pool. An important characteristic about the Mendelian population is that they are breeding isolates, although they are isolated to varying degree. All individuals of the populations are not equal in their ability to survive and reproduce. The species can be viewed as the logical Mendelian populations. Species is a term coined by the John Ray in 1693. It forms the basic unit of the biological classification. With reference to the taxonomy, species refers to the morphologically distinct and reproductively isolated natural population or a group of populations wherein the member of the species interbreed freely among themselves, not with the members of the other species, and form a genetically closed system, share the common gene pool. There would no longer be a distinctive kind of the organisms if species interbred among themselves. Phylogenetically, a species caters to a lineage distinct from other such lineages, perpetuated ancestors to the descendants over time. Biologists and taxonomists have tried to define these species from the several scientific avenues, starting from the morphologically and moving towards the genetics. This table provides us the glimpse about the various species concept as given by the Campbell et al. And Coyne et al. Or Coyne and or in 2004. Now let's understand the concept of the speciation and the process of speciation. Speciation is a term coined by the orator F. Cook in 1906. It is an evolutionary process related to the formation of the new biological species from the pre-existing ones through genetic modifications. This evolutionary process initiates when the member of a particular species become isolated from each other and starts to evolve independently. White emphasized that speciation is a crucial evolutionary mechanism by which the organisms adapt so as to exploit the diverse environmental conditions available to them. Charles Darwin mainly concentrated to demonstrate the evolutionary changes by the action of a natural selection and discussed how population adapt to their environment through the process of the natural selection, but not how this adaptation paves the way to the formation of the new species. Most critics of the Darwinian theory of evolution emphasize that the genetic variations can occur within a species as the result of the selection. They maintain that such variations is bounded by the genetic limits of that particular species Thus, critics of the Darwin theory of evolution views variation within the species as a difference in the degree, and variation between the species is perceived as a difference in the kind. Breeding isolates are developed in situations where a breeding population does not share a common gene pool, and if these isolates are subjected to the different type of the selections. Speciation takes place if mating barriers prevail. The resulting breeding units or breeding isolates will initiate to formulate their own distinct gene pool. Hence, the degree of the genetic dissimilarity between the breeding isolates is intensified by the prevalence of the different environmental conditions and different processes of the selection. Thus, speciation will be more likely to occur. The population will evolve local differences in the gene frequencies as they are isolated by a reproductive barrier. Also, the process of speciation is greatly speed up if genetic isolation of the population subgroups is complete. According to Pawson, who highlighted that processes such as the geographical separation, differential selection or new genetic material from the migration tend to increase the genetic variation within a species and favor the formation of the breeding isolates, while processes such as the panmictic or random mating decrease the genetic variation within a species by promoting the gene exchange between all segments of the species. Both the fossil record and the degree of the genetic similarity between two species can aid in assessing how long ago the speciation occurred. Two major processes related to the speciation are the anagenesis and the cladogenesis. They are observed in the course of the human evolution. Figure 1 shows the cladogenesis and anagenesis as given by the Pawson. Anagenesis or the phyletic transformation is an evolutionary process which implies that time plays a crucial role as an isolating factor between the members of a lineage. In other words, evolutionary transformation can occur in a species over time to a sufficient degree such that their later organisms could eventually be treated as a different species. This type of speciation relates to the progressive adaptation to a fairly stable environment. In contrast to the anagenesis, cladogenesis relates to the branching evolution, that is, it occurs when an ancestral species subjected to changing environment conditions splits or branches into several distinct or daughter species, each occupying its own habitat. This speciation process is quite common in the fossil reconstruct. The number of the available ecological niche influences the rate of the speciation. In certain cases, a population of the one species dispersed throughout a geographical domain and each acquires a distinct ecological niche over time. This varied demands of their new lifestyle leads to the subsequent branching of the single ancestral species. This process is termed as the adaptive radiation. As a sign of the adaptive radiation, the fossil records exhibit the relative sudden proliferation of the related species. Figure 2 shows that the Honeycreeper bird exhibit adaptive radiations as from one original species of bird, multiple other evolved, each with its own distinctive characteristics. Darwin's finches offer the typical example of the adaptive radiations. As of the present, there are 14 distinct species of the finches on the Galapagos Island, and all of them have evolved from the one ancestral species, which migrated to the island only a few million years ago. Thus, from the ancestral seed-eating species, several different species with the altered beak arose, facilitating them to become the insectivorous seed-eaters and the cactus eaters. Now, let's understand the mode of speciation. Evolutionary biologists have outlined several modes of speciation. For example, J.S. Huxley in 1942 emphasized three crucial types of the speciation, namely geographical, ecological and genetic. The M.J.D. Whites suggested that the three main sets of variations are engaged in the speciation processes, which are genetic mechanisms generating the genetic variability, genetic isolating mechanism leading to the origin of reproductive isolation, and the geographic component ranging from the complete or allopatry to absent, that is sympatry. He outlined 7 models to illustrate the mechanism of speciation. According to White, the number 1 is the strict allopatry without a narrow population bottleneck. Number 2 is strict allopatry with a narrow population bottleneck, that is founder effect. Number 3 is the extinction of the intermediate population in a chain of races. Number 4 is clinal speciation. Number 5 is area effect speciation. Number 6, Stacypatric speciation, which is primarily chromosomal. Number 7 is Sympatric speciation. The first 3 models represents the allopatric or the geographical speciation, and the next 3 relates to the semi-geographic speciation. Allopatric, Sympatric, Parapatric and Stacypatric are the prominent mode of speciation to illustrate the process in animal species. Figure 3 shows the different type of mode of speciation. Now let's talk about the allopatric mode of speciation. This figure shows the allopatric speciation model as given by the Jenkins in 1990. Allopatric, where allo means other and patric means a homeland, speciation is also referred to as the geographical speciation because it requires the physical separation due to distance or geographical barrier at initial stages. It takes place where a main ancestral population is divided by some sort of barrier and separated segments of the population adapt to different environments. These separated segments of the population experience genetic drift as barrier inhibit the gene flow, which acquires the mutations and formulate a distinct gene pool separate from the ancestral population. The genetic difference between the newly separated population intensifies the longer they remain isolated. Even if there is some gene flow after the members of the two populations initially come back into contact, character, displacement and reinforcement may amplify the initial differences between the population and lead to the formation of two new species. There are two main subtypes of the allopatric speciation, that is dichopatric and peripatric. The dichopatric model did not assume the founder principle, but in peripatric, the speciation model, there is an involvement of the founder principle. Allopatric speciation is exhibited by several cases such as the evolution of the Darwin's finches on the Galapagos Island, evolution of the land snails of the genus Patula in the Society Island, and locusts and mammals on the various islands. Now let's discuss about the sympatric mode of speciation. Bush in 1975 defined the sympatric speciation, where sym means same and patric means the homeland, speciation as the origin of a new species characterized by the reproductive isolation within the dispersal area of the parental species. The sympatric speciation is the evolution of the reproductive isolates without a geographic isolation, and this was according to the Meyer given in 1963. This figure shows the sympatric speciation model suggested by the Jenkins Singh in 2012. Singh stated that sympatric speciation involves the instantaneous appearance of the reproductive isolations between the segments of the same population, and new species originate in the same geographical area. According to the Coyne and Orr in 2004, there are 4 criteria which have been outlined to infer that a particular case is best illustrated by the sympatric speciation. Number 1, the species thought to have arisen via sympatric speciation must have largely overlapping geographical ranges. In principle, sympatrically derived species could become allopatric over the time, but it is not clear how to demonstrate the such secondary allopatry. Number 2, speciation must be complete. We cannot declare a case of the sympatric speciation if speciation has not occurred. In practice, whether or not speciation is complete is a taxonomic decision that is contingent on the definition of the species, that is the biological species concept according to the Cohen and Orr case. Number 3 is the clades throughout to arise via sympatric speciation must be sister species or morphophyletic groups, that is, we can support the sympatric speciation only when the evidence has not been obscured by the subsequent non-sympatric diversification. Number 4, according to the Cohen and Orr, the biogeographic and the evolutionary history of the group must make the existence of an allopatric phase very unlikely. This is not so much a criteria as a statement that the first 3 criteria are necessary but not sufficient in their views to reject the alternative to the sympatric speciation. These criteria clearly relate to the biogeographical concept of the sympatric speciation. To infer the sympatric speciation under the population genetic concept, an additional condition must be met. That is, the evidence must support the panmixes of the ancestral population. Now let's understand the parapatric mode of speciation. This mode of speciation emphasizes that selection alone is not sufficient to form a new species, and new species can be formed if the selection works in association with the partial genetic isolation. There is no specific extrinsic barrier to the gene flow. A particular species occupies a wide array of the environment, incorporates different morphological and behavioral features in each of these environments, and this variation may cause the particular species to vary in the different environment. Under such situation of slight distributional overlap, such as of habitat boundaries, members of 2 different species having different characteristics may mate and produce the hybrids. Singh in 2012 highlighted that parapatric species may lack the behavioral isolation and hybridize, but their hybrids are sterile. Andler in 1977 suggested that isolating mechanism develop and intensify in a cline along an ecological escarpment until the 2 adjacent populations are finally introducing isolated selection inhibit the mating between the members of population from different habitat, and as a result gene will be reduced and finally 2 new reproducing isolated species will evolve. Now let's understand the Speciatic mode of speciation. The Speciatic mode of speciation is also termed as the classic chromosomal speciation model, as given by the White et al. In 1967 and White in 1968, because new species arise sympatrically, initially by the chromosomal rearrangement within the geographical range of the parent species, and the new population disperses and proliferates within the range of the parent species, spreading by the parapatric distribution. This mode of speciation is characterized by 2 principal features. Number 1 is the chromosomal rearrangements which produce the barrier to gene flow between the parental and the daughter chromosome types due to the meiotic abnormalities in the chromosomal heterozygotes, and number 2 due to the spread of the new chromosome types from their point of origin into the distribution of a parental chromosome type occurs without the geographical isolates, leading to parapatric distribution of the chromosomal races. Now let's understand the isolating mechanism influencing the speciation. The mechanism of the reproductive isolation refers to an assemblage of the evolutionary mechanisms, behaviors and physiological processes that allows the isolated groups to evolve into distinct population or species necessary for the speciation event to occur. These reproductive barriers inhibit the mating between the members of different species or ensure that if mating occurs, the offsprings are non-viable or infertile. Hence, reproductive barriers guard the integrity of a species because these barriers restrict and inhibit the gene flow between the related species. Scientists have classified the mechanism of the reproductive isolation into two major categories, that is pre-zygotic barriers or the pre-mating isolation and the post-zygotic barrier, that is post-mating isolation. The pre-zygotic barriers refers to the mechanism that inhibits the reproduction from taking place. This includes the barriers that prevents the fertilization when organisms attempt the reproduction, and the post-zygotic barriers occurs after the zygote formation, which ensure that if the mating occurs, the product of the mating does not produce a viable offsprings. Pre-mating isolation mechanism includes the temporal or differences in the breeding schedules or ecological, that is differences in the ecological or the habitat preferences. It can be mechanical also, where difference in the genitalia in the animals is there. Physiological, because on the basis of the adverse physiological reaction such as the non-viability of the sperms in the reproductive tract of the female of the other species, and it could be behavioral because of presence or absence of a specific behavior. Isolation and post-mating isolation mechanisms incorporates the gametic incompatibility, that is the non-fusion of the gametes. It can be zygote mortality, where zygote dies soon after the formation. It could be hybrid inviability, where hybrid dies before attaining the sexual maturity. Now let's understand the macroevolution, the phyletic gradualism and punctuated equilibrium. Macroevolution is primarily based upon the describing evolutionary patterns which we have discussed earlier at the species level. The macroevolutionary studies focus on the change that manifest at or above the level of the species, and in contrast, the microevolution relates to the evolutionary changes within the species. Mutation, genetic drift, isolation, which could be natural, sexual or the social, selection and the hybridization are the prominent microevolutionary forces which lead to the evolutionary changes in the terms of changes in the gene frequency. It has been emphasized that both the evolutionary processes are related to each other, as they do not imply any difference in the underlying agencies and the aggregate effect of the microevolution during the course of lifetime in the long term leading to the macroevolution. Macroevolutionary studies are centered upon the examination of the fossil records. There are two prominent models related to the tempo and the mode of macroevolution. The first emphasizes that the microevolutionary processes alone can sufficiently explain the grand patterns and the radical changes in the tree of the life. This model suggests that the speciation may occur through the gradual changes within the lineage, such that over the time species is transformed into other species. In contrast to the first model, the second model proposes that the grand patterns in the history of the life cannot be explained solely on the basis of change in the allele frequencies over the time, even rapid ones. Instead, scientists propose that large changes on the tree of the life were preceded by the events that decoupled the tempo and the mode of evolutionary changes from predictable microevolutionary processes. Thus, in the first model, changes is slow. In the second, speciation events are rapid and interspersed among periods of no change or stasis. The first model has been termed the gradualism and the second is referred as the punctuated equilibrium. Gradual and the punctuated equilibrium are two ways in which the evolution of a species takes place. Punctuated equilibrium and phyletic gradualism are contrasting patterns of evolution among a spectrum of patterns observed in the fossil record. In punctuated equilibrium, species tend to exhibit the morphological stasis between abrupt speciation events, while in the phyletic gradualism, species undergo more continuous changes. The central theme of the punctuated equilibrium includes three concepts: stasis, punctuations, and dominant relative frequency. Stasis refers to a long period of relatively unchanged form. Punctuation is a radical change over a short duration, and dominant relative frequency is the rate these events occur in a particular situation. According to the Silo et al., punctuated equilibrium emphasizes that number 1, evolutionary change is connected in speciation events. Number 2, most species remain pretty much the same once they have come into being. This lack of substantial change over millions of years is called the stasis. Number 3, speciation events are normally confined to the small populations, peripheral isolates that have become separated from the bulk of the species. As these isolated populations are small and transient, we should not expect to find them in the fossil record. This figure demonstrates the prominent differences between the phyletic gradualism and punctuated equilibrium. Stippling is used to denote the different species. The morphological variability within the population at any point of time is represented by the belt curves. According to Conroy in 2005, this table provides an account of the major differences between the phyletic gradualism and punctuated equilibrium on the basis of the rate of phenotypic changes, the direction of the phenotypic changes within a species, the population size for the speciation, whether the speciation is purely a function of the time, how new species arise and the implications for the species. Now let's summarize this module. The concept of the evolution is central to several biological concepts and ideas. Evolutionary transformation leads to the formation of the new life form from an ancestral one. It relates to the adaptation of the organism to their dynamic environmental conditions and can lead to the altered genotypic and phenotypic framework and even the formation of a new population and species. Species relate to the basic unit of the evolution. Evolutionary biologists have made several attempts to provide the fundamental understanding of the species on the basis of different diagnostic criteria ranging from the morphology and moving towards the genetics. Several prominent species concepts have been proposed: biological species concept, phylogenetic species concept, evolutionary species concept, ecological species concept, and many others that specify how species boundaries can be drawn in practice. As gene exchange between the population is inhibited owing to the evolution of the isolating mechanism, new species eventually arises. This relates to an important evolutionary process called the speciation. The prominent speciation models are the Allopatric, Sympatric, Speciation, and Parapatric speciation. Evolutionary biologists have emphasized that the reproductive isolations mechanism that restrict or prevent the gene flow can occur in a variety of ways. Isolation mechanisms have been broadly classified into two groups, that is pre-zygotic barriers and the post-zygotic barriers. Speciation rate is influenced by the two prominent models of the macroevolution, that is gradualism and punctuated equilibrium. The first model of the gradualism is characterized by the slow change that leads to the gradual and uniform transformation of one species into a new species through anagenesis. In contrast, The second model, that is the punctuated equilibrium, is characterized by the rapid speciation event, which are rapid and interspersed among the periods of stasis for no change. Thank you.